Part 7 of Chapter 5 goes into another type of genetic variation, which is looking at chromosomal mutations. So far the types of genetic variation that we've looked at were new alleles and new genes, and now we will look at larger scale changes that occur with either entire sets of chromosomes or large sections of chromosomes. So when we talk about these chromosomal mutations and how they might differ from the ones we've already seen, so far we looked at small changes that were typically looking at point substitutions and single base pair, possibly very small insertions or deletions, but these things that would be very small in scale. When we look at chromosomal mutations, we're often looking at things that you could actually see when you looked at the chromosome, because you could actually see length differences, or you might see some structural changes like a loop in the chromosome and that sort of thing, or you might see an extra chromosome, or you might see a whole nother set of chromosomes. So here we're looking at things that are much more large-scale changes to the genome. Many of these we could actually see by looking at the chromosomes. So when they talk about this gross morphology, that's really what they mean. If you could stain chromosomes and look at them at high enough magnification, you would actually see differences in those chromosomes. Whereas a simple base pair change, you're not going to see those when you look at them. Some of the changes that we're going to see might not really change what we would see. It may not be like a whole missing piece of a chromosome or an extra one. It might actually only change the gene order. And then others might actually be large sections of a chromosome missing or added, or it also could be entire sets of chromosomes deleted or added, or just a single chromosome added or deleted. When we take a look at these chromosomal changes, we also are not going to look at all of the different types. When you study this kind of mutation in genetics, you go through a lot of different types. You go through aneuploidy, different types of polyploidy, translocations, large-scale insertions and deletions. We don't look at all of those here, primarily because they probably don't have big effect on evolution. Many of those kinds of things result in severe complications to the cell that don't get passed down over the long evolutionary time scale. So evolutionary biologists don't focus a whole lot on some of those because we don't tend to see them persisting. They primarily here are just looking at the ones that are really important to evolution in a big way. One of these is inversion, and then the other is genome duplication. So when they look at genome duplication, they're typically looking at sets of chromosomes being duplicated. When we have an inversion, the chromosome is breaking, so we basically get two nicks in the chromosome here. Here's what our chromosome should have looked like with A through F genes lined up, but what ended up happening is it's formed sort of this little loop, and then it broke and those arms reattached, but the way that they reattached, if you look at it now, the genes are actually in a different order. Now instead of C, D, and E reading left to right down here, they've flipped over, and so now they're in a different order. So it changes the ordering of those genes. And when it changes the order of the genes, it also is going to affect linkage. Because remember, linkage between two genes is a function of the distance between those two genes. Before, we had a certain distance here between B and C were pretty close together, and they might have been inherited together more often than not because of their proximity. But if I take C and I move it further away, now there's a good chance that I get a crossover somewhere within that space, because the more space you have here, the more likely you are to get a crossover and more likely to get reshuffling of genes. If you have a very short space here, you typically don't get a crossover, and then you don't reshuffle those. So if one parent has a big B and a big C and they don't reshuffle, those tend to go together. The idea is if I rearrange those, I'm changing that linkage. So now it may be that B and E tend to go together more often as an inherited group as opposed to B and C. The other thing that happens with an inversion is when you have this inversion, now when this particular chromosome tries to line up with its homologue, to get these to line up, they form what's known as an inversion loop. so that they can line up correctly. And then once it forms that loop, they tend to not be able to cross over. So essentially, an inversion reduces the amount of crossover that you're going to get. Let's imagine now that because there can be no crossover here in between E, D, and C, these are essentially acting like one big gene because we can't reshuffle them. And so it creates what we sometimes refer to as supergenes. These sections of multiple genes together that get inherited as a unit because we can't break them apart because we don't do a crossover in that inversion. We'll look at an example from Drosophila. These populations of Drosophila are polymorphic for at least one inversion. And what it means to be polymorphic is that some individuals in the population have chromosomes that do not have the inversion, and then other individuals in the population do have the inversion. So you can think of the inversion almost like a trait. Some individuals have the trait, some individuals don't have the trait. Each one of these data points is from a population asking the question, in that population, what is the frequency of the inversion? And so if the frequency of the inversion was 0, then none of them have the inversion, right? And if the frequency of the inversion was at 100, then all of them would have the inversion. In this particular case, we do have individuals that have it and individuals that don't. So there's polymorphism there. And so we're looking at these different populations. What is the frequency of the inversion in that population? And then we're correlating it with latitude. If we increase latitude away from the equator, so we're moving away from the equator in the southern direction, or we're moving away from it in the northern direction, either way, what we see is a nice trend. We see a nice correlation between changes in latitude and the change in this inversion frequency. If we see a pattern like this, and particularly when we see it being repeated both in the southern and northern hemisphere like this is telling me that this is probably not a random event, that there's something causing this, because we wouldn't expect to see this nice of a pattern with something that was just generated randomly. It's probably telling us that selection is favoring certain levels of polymorphism for this inversion along this gradient. What it would suggest to me is that at this lower latitude, when we're closer to the equator, there is selection favoring lower frequency of this inversion in the population. As we increase in latitude further away from the equator, we're seeing that this frequency increases. So selection probably favors this inversion here more so than it does down here. So something about latitude is favoring that inversion. When you see latitudinal changes, one of the first things that should pop in your head is that it's probably something that is being driven by climate, because that's one of the biggest things that changes when you change latitude, is that the climate changes. You end up going from an area that essentially has no seasonality to it, is pretty much the same all year round, to an increase in that seasonality, obviously changes in precipitation as well, and temperature that goes along with that. This pattern that we just looked at, where you see a regular change in the frequency of the allele over a geographic area, in this case it's a latitudinal change, but we could be going up the side of a mountain. It could be an altitudinal gradient. So it can be really sort of any type of this gradual change that we're going to see. We refer to this as a cline. This cline that we see with respect to this inversion frequency is suggesting to us that natural selection is operating on that inversion. It's making the inversion favorable at these higher latitudes and not as favorable at the lower latitudes. One of the things that we see is that this cline is also correlated with gradual changes in the climate from a kind of a wet marine environment to more Mediterranean climates to more desert and drier habitats. And so we're seeing, as we see this change in the fruit fly population and this inversion, we're also seeing that there's that climate change going along with it. So again, this evidence of a cline that we see here is telling us that this has probably been under selection. This is probably not just some historical accident that created this pattern, but that selection has favored more of the inversion here and fewer of the inversions back here. So next we come up with genome duplication. So that's the next big thing in evolution as far as chromosomal changes go. So now what we are looking at is getting entirely new sets of chromosomes when we look at genome duplication. Now, in genetics, you looked at sort of a middle step here, which would be looking at the duplication of maybe only one of the chromosomes of a set. In some types of Down syndrome, you have one extra chromosome, number 21. And so that's not a genome duplication per se. That's just a one single chromosome being duplicated, not the entire genome. So that individual has 47 chromosomes instead of 46. For a genome duplication, say in humans, we would have to go to 92 chromosomes. So it would have to be a multiple of the entire set of chromosomes. If we have only one set of chromosomes, we've seen that term before. That's what gametes are, and we call it haploid. If we have two sets of chromosomes, which in many organisms is what we see in the adult stage, is the diploid condition. If we have more than that, if we have more than two sets, we typically refer to it as polyploid. And we can give it names. We can call them triploid or tetraploid, and we can specify how many sets they have. But typically just this general term polyploidy would be anything greater than two sets. We see polyploidy quite a bit in plants, but not very often in animals. One of the reasons why that is probably true is because plants can self-fertilize. The problem with an animal becoming polyploid is when we get fertilization, if we don't have the same sets of chromosomes, the same numbers, typically fertilization doesn't work. That's one of the reasons why species tend to be separate from other species in most cases, because they have a certain number of chromosomes, and if they do try to interbreed, they're sterile because of the mismatch of the chromosome number. So having chromosome number match is really important. If you were to start producing gametes of a different chromosome number, you wouldn't have anyone you could mate with unless somebody else is also making those gametes with that different chromosome number. And so that doesn't happen very often in animals. But plants have this really unique ability to self-fertilize. So a lot of plants, they have both male and female flowers in the same individual. So they're essentially like hermaphroditic organisms. And so they can actually self-fertilize. So if they do increase or decrease their chromosome numbers, they can still reproduce by fertilizing themselves. That's probably one of the reasons why we see it a lot more in plants than we do in animals. There's a couple of reasons why polyploidy is probably very significant to evolution. One is that if we think of a plant that undergoes this duplication of its genome, and now it has a whole extra set of chromosomes. So let's say it was diploid and now it's triploid. Can't very easily breed back to individuals that don't have the same chromosome number as you. This could lead to immediate speciation. Polyploidy can lead to very rapid speciation because individuals that are polyploid now can only mate with other individuals that are polyploid, and so whatever their parental species they came off of, they often can't mate back to that original. Once we get this genome duplication, think of what it does for adaptation. We talked before about how gene duplication gave rise to a lot of new abilities for organisms to create different gene families that would then be able to produce slightly different forms of a gene, slightly different forms of a protein, so organisms could adapt to different conditions. Well, if that's true for increasing the gene number, imagine when we go and duplicate entire genomes. Now we have a whole bunch of new genes that we can use that might would allow this organism to adapt to a wide variety of new conditions. Here are some data from yarrow plants. What they're looking at over here are sets of chromosomes. So here's a yarrow plant that has 4 sets of chromosomes. Then what they did is they created a hybrid, and this hybrid individual has 6 sets of chromosomes. And they call this the Neo6. They created it by taking a 4X and a 2X, and they originally created this 6X. But the next one, the darker green one here that is the 6X, it is by crossing other 6X. So we have our first generation where we created it by hybridizing, but now we're just going to take that population and continue to propagate it. What we're seeing over here is the change in the survivorship as we increase the size of that genome. As we increase chromosome number, we see a drastic increase in the overall survivorship of those individuals. So it would appear that having more chromosomes and having a larger genome really helped those individuals survive. And we do also know that this was done in a situation where the plants were stressed in a very dry environment. So if they're in a dry environment, which again, expanding your genome may allow you to go in and use habitats and new environments in a way where the original plant could not. We see that survivorship has increased when we increase the number of chromosomes. Remember what natural selection is. Natural selection is differences in the ability to survive and reproduce. So it looks like this hexaploid definitely has a selective advantage because of its survivorship. What they're doing in part C over here is also bringing in the second piece of natural selection, which is differences in their ability to reproduce. Look at the expanded flowering dates that you have for that hexaploid individual. Your tetraploid individual was very isolated into flowering in a short time frame in sort of late June to early July. And now this hexaploid can start flowering much sooner and has a much longer time frame for flowering. So one can only assume that that would potentially and also increase its reproductive ability. So now we see increases in its survivorship, and we also see potential increases in its ability to reproduce. So they're probably better adapted in particular environments. In this case, probably these dry environments, they do much better than, say, the original tetraploid species that they came from.